actively contributing to a wide range of areas from lab projects to national projects, including the first human genome sequencing project in the university and the SARS-CoV-2 genome sequencing project, etc. So if you are ready, please start, Yoshinori-san.
Okay, good afternoon, everybody, and thank you for kind introduction. And also, I'd like to thank you for organizing this great event, and thank you for giving me the opportunity to talk about this talk today. I'd like to talk about solution and new challenges in accurate long-read error. This is agenda of today. I'd like to introduce what is cows and what is owl now first. Then I'd like to go to briefly background and all challenges. Then I'd like to introduce recently published quick workflow for plant genome acquisition. We name it this workflow WIFO. And also on the top of that, we are having kind of emerging project in our institute. I'd like to share some of new insights and kind of background what's happening in Middle East. And as usual, I'd like to conclude my talk with summary. So, where we are. Maybe you can see, is there any window? I don't know, it's just pointing it. So, I think Asia is quite good place, quite greenish. And as you can see, some area is more yellowish. And so, for example, kind of demand in the society is actually quite different. So, for example, in Middle East, agricultural research project is quite popular, like sequencing of plant genome and animal, like cattle or camel. And location of Saudi Arabia, cows is west side, west coast of Arabian Peninsula. We are facing so-called Red Sea. It's a beautiful sea area. And I think now, World Cup is happening, and famous Qatar is actually located the other side of Arabian Peninsula. And the famous Dubai is here. I often say, Yoshinori, good luck in Dubai, but actually it's not. It's the other side of Arabian Peninsula. So quite far away from Asia, but as you can see, it's a quite different climate, so different research lab is available. So we are quite also young university. The university itself was established in 2009, and we still developing lots of things. But say we are also quite international environment. So people coming from more than 100 countries. And out of university, for example, common language is Arabic, but inside our campus, we use English mainly. And population is say around 7,000 for now, but it's keep expanding. We, Bioscience Core Facility is actually kind of de facto sequencing hub in the nation. So we need to deal with lots of demand in the nation with current scientists in our lab. So before going into detail, I'd like to briefly talk about what is accurate long read. I think most of people know what is NGS short read data. It's quite massive scale and very good at changing dynamics. The shortcoming of this type of data is, of course, it's short. And long read technology represented by PacBio or Nanopore actually try to improve this as a barrier. They try to seek more and more region, but shortcoming of this technology is accuracy. So usually long read technology is famous for error-prone dataset, like 10 to 5%. But recently, there is a new game changer provided by PacBio, and it's actually quite accurate. And I'd like to briefly talk about this point. And this kind of data is called accurate long read. but it's quite different in terms of accuracy from even long-read technology. So I'd like to bring all challenges in case of typical de novo assembly, because long-read technology is famous for its error-prone characteristic. Error correction was a major step. For example, this is a typical assembler, a kind of standard assembler called canoe. Because of high error rate, usually before assembly, lots of error correction need to be done. This means mapping all versus all. And so this can be a quite time-consuming step, and usually weeks or sometimes months on cluster was needed. So bottom line is slow, and sometimes, of course, genome assembly is a big deal, but this was a major headache for, I think, body marshals. So lots of human intervention or applying manual marchable technology was needed. So this is kind of old product made long time ago. This is first generation. X-axis is length and Y-axis is accuracy. The first generation can be quite accurate, but it's unfortunately low throughput. This is Illumina, short, but it can be quite massive scale measure and okay accuracy. As you can see, third generation long read data has much lower accuracy, but it's longer. But this scenery is changing. In around 2020, PacBio developed a new technology called Hi-Fi, and this keeps increasing accuracy. And now it's at least as accurate as Illumina, and it keeps length. But now we have technology having both accuracy and length. And maybe Nanopore was a bit upset about this development, so they taking length. So Nanopore can achieve even much longer, say longer than 100 kilobits per. This is also happening. But this is kind of technology at glance. This is a bit old, benchmarking result done in our lab. So this is Nanopore. Accuracy is say Q10, like 10% error rate. And Illumina, we use so-called ring-to-read technology, kind of virtual long read. But this sometimes struggle with accuracy and like say Q20 region. But as you can see, PacBio Hi-Fi can achieve almost Q30, so around 0.1% error rate, keeping 20,000 base pair. This is kind of background happening in the community, and now challenge is how to use this kind of new data set. So because of this advancement, we established a study kind of project named LeafGo. And the point is to answer the demand in the community, quickly develop genome from this. So for the project, we chose multiple plant species. One is eucalyptus, because this has deployed a kind of standard approach. And also genome size is relatively small, around 500 megabits per. And on the top of that, we chose a bit more challenging plant species, peanut, because this genome has so-called allotetraploid. Roughly speaking, this has two diploid genome and two subset of genome within it, having some level of similarity to each other. And the genome size is 2.6 gigabase per. It's much longer than eucalyptus. But this is kind of a very simplified summary of the result. This project was done by talented scientist Salim and Kamel in our team. So these left two bar bars shows accurate long read, and this right two bar bars shows long, old long read technologies, more erroneous. This made y-axis is so-called N50. Simply speaking, the longer, the better. So as you can see, if we use accurate long technology, it can be much longer, almost double size, if compared to old technology. And more remarkable stuff, at least for us, is computational requirement. Y-axis is CPU hours in log scale, so say more than 1,000-fold reduction of computation can be achieved. Now, at least for eucalyptus case, actually we could assemble genome within 2 to 3 hours on our desktop computer. So because of this demonstration, we could establish workflow from leaf to genome acquisition roughly within 1 week. And we named this workflow LeafGo. And this was published in Genome Biology. So this is surface plot. So LeafGo can produce nearly chromosome level assembly within, say, a couple of hours. These are reference genome of mentioned species. There are some exceptions, but mainly most of chromosome can be covered by only one counting. So if we use accurate long-read technology, within 2 or 3 hours, we can get this level of high-quality draft genome nowadays. This is peanuts. Peanuts is more challenging, so unfortunately a bit more fragmented, but still most of reference chromosome can be covered, say 2 or 3 counties. And what we noticed during the project is, say, this accurate long read is very promising, but sometimes there is a drawback. This is a specific region of chromosome 2 of eucalyptus genome. As you can see, this region has 0 coverage. So if we don't know the other data, like nanopore sequencing data, this looks like deletion. But actually, this is not deletion. This is coming from sequencing bias. So this kind of, this is not popular, but this kind of drop can happen. Maybe this is good to know. It's a bit difficult to explain why this happens, but there is interesting 45 insertion in other technology, but it's not observed in hi-fi technology, accurate long read. And some people mentioned some specific sequencing signal may hinder sequencing using this packed bio hi-fi. So sometimes please keep in mind this kind of dropout can happen still. So summary so far is we could demonstrate now at least diploid or some level of protein genome can be assembled very quickly if we use accurate long read. The challenges of this project is because we do not have parental information, so we could not assess how each haplotype is contiguous. So this lead to new project. It's going on. So let me share some kind of ongoing preliminary results. Title is a bit big, but this is our motivation. We want to get as many as variant from one single individual. As you know, there are many types of different variants within our genome. SNP and insertion, deletion, regardless of size. And of course, tandem repeat is important, copy number variation. But there are many approaches suggested to tackle detection of variant. Let's say one promising approach could be using long read. If so, we can spawn even relatively long structure variant. But we don't know how good this technology, so we want to assess. And assessment strategies, because we need to get information for haplotype, so we try to get parent and kid. So we actually look for kind of this type of parent kid demand, and we found actually this social issue in the Kingdom of Saudi Arabia because of cultural background. Excuse me. So-called consanguineous marriage is quite common in the nation. It's kind of marriage between, say, two cousins. And actually, I was surprised. So, for example, this kind of marriage rate in the kingdom can be even higher than 50%. So, if, say, some potential allele existing parent, for example, autosomal recessive disease can be quite serious, and they actually demand to analyze this type of data in the kingdom is quite high. So we actually working with actual trio potentially having autosomal recessive disease, and for clinical perspective, and also we want to assess our technology using this kind of setting. And, because clinical project goes to too many details, genes, and this is bioinformatics conference, so let me focus on typical HG002 case using new technology. So if we have, say, 35x coverage of accurate long read, as you can see, as we discussed, assemble can be done quite quickly, and a problem is how accurate the assemble genome is. This is accuracy, so roughly Q50 to Q55 accuracy can be achieved using just one technology. And so-called switch error. Switch error is kind of error between two haplotypes. But sometimes assembler can mistakenly